Hey all, welcome to Learn It. Today we are going to solve numericals related to the chapter Molecular Biology. So let's get started. So what are the uh, prerequisite concepts which you need to know before solving any numerical in this chapter? So the first one is you need to know the distance between two nucleotide pairs, which is 3.4 angstrom. Here every unit, like the unit of measurement I have used is angstrom, and if in the question they've asked in nanometer, you need to know the conversion. So we all know that 1 angstrom is equal to 10 to the power minus 10 meters. So which you can write is 10 to the power minus 9 into 10 power minus 1 meter. है ना? Now 1 angstrom would be, because you know that 10 power minus 9 meters is equal to 1 nanometer. Yes, 10 power minus 9 meter is equal to 1 nanometer into 10 power minus 1. So basically 1 angstrom is equal to 10 power minus 1 nanometer. Alright. So with this idea, you can convert any measurement which is given in angstrom into nanometers. So sometimes in the question, they can also give you as the rise per base pair is equal to 3.4 angstrom. Similarly, the distance between two nucleotide pairs can also be given as between two successive steps, okay? Alright, so whenever they ask you any question related to distance between two nucleotide pairs, you'll have to know that it is 3.4 angstrom. The next idea is pitch of the helix. The one complete turn is called as pitch, right? As you can see in this diagram, this is your pitch. This is from the NCERT diagram. So one complete turn is your pitch. Alright, so which is 34 angstrom. Alright, so basically how do you know this? Every turn, like the rise per base pair is 3.4 angstrom, right? And how many rises do we have? We have 10 rises. We have 10 rises, which you know per pitch you have 10 rises. So 10 multiplied by 3.4 angstrom would be 34 angstrom. So the pitch of the helix is 34 angstrom, or otherwise they could also tell you as length of one loop of bDNA is equal to 34 angstrom and length of one complete turn is 34 angstrom. Alright, so this is the second concept related to the pitch of the helix. Moving on to the third type of numerical, length of the DNA. So whenever they're going to give you the length of the DNA, so you should remember this thing, the total number of base pairs which is present multiplied by the distance between two consecutive base pairs, which is very obvious. So let's say for example, just a rough sketch. So what would be the entire length of what would be the entire length of this okay, I'll draw this here. What would be the entire length of this whole ladder, given that the number of base pairs is given and the distance between two consecutive base pairs is also given, okay? Now if they ask you to find the length of the DNA, what will you do? You will multiply this distance with the number of base pairs to get the entire length. Yes. Similarly, the length of the DNA is what? Total number of base pairs multiplied by the distance between two consecutive base pairs. Alright, and what if they ask for, like, how do you find the total number of base pairs? So total number of base pairs, just remember this formula, okay? You're clear with this, right? Total number of base pairs multiplied by distance between two consecutive base pairs. In the question, they may not directly give you that the total number of base pairs is this much. Rather, they would give you as number of nucleotides in one of the strands. Because if the number of nucleotides in one strand is given, the same number of nucleotides will be there in the other strand as well. So instead of giving the total number of base pairs, they'll give you as number of nucleotides in one of the strand. Alright. And you also know that one base pair has got two nitrogen bases. Yes. So this is one base pair. Alright. These two together constitute one base pair. Whereas If they give you as number of nucleotides in one strand, they mean only this thing. Only this they mean. Alright, so one base pair would be equivalent to 2 nitrogen bases. So therefore, the number of nucleotides in one DNA will be what? Twice the number of base pairs. Yes, so this is one base pair. This is what? One base pair. And how many nucleotides do you have? You have 2 nucleotides. Sorry, two nucleotides, right? So, number of nucleotides in one DNA, will be, number of nucleotides in the entire one DNA would be what? Twice the number of base pairs. Okay. For example, if they give you that there are 8 turns, okay, if there are 8 turns in a DNA, then what does it mean? Like 8 turns having 80 base pairs, they say. Then how many nucleotides would you have? You'll have 160 nucleotides. Yeah. As I told you, this is one base pair, whereas this is one nucleotide. So if there are 80 base pairs means there are 160 nucleotides. Alright. So this is the third concept. So this could be a bit tricky if you don't understand, but just try and understand yourself. Okay. So everything is clearly explained in this diagram. So this is what? This together constitutes one base pair. Yes. And this, let us suppose this is A. Okay. One nucleotide would be, this entire thing is one nucleotide. Similarly, if this is T, okay, adenine base pairs with thymine, so this entirely is your other nucleotide. So 2 nucleotides, where is one base pair? Okay. Let us move on to the fourth concept, which is very commonly asked. So Chargaff's equivalency rule. So according to Chargaff's equivalency rule, in a double-stranded DNA, the amount of purine equals the amount of pyrimidine. Okay. So A plus G is equal to T plus C. Just remember this. Alright. Purines is equal to pyrimidine. Okay. One more concept is that A plus T, this is what A plus G by T plus C is, like A and G, and both of them would be equal. And you know why? Because adenine always base pairs with thymine and guanine base pairs with cytosine. Yes. So obviously if ATGC, yeah, so this and this would be equal, right? Only then it can form a double helix DNA. Alright. So moving on. Let us now solve a question. And whenever like they give you the base sequence of one strand is given and the sequence of complementary strand is asked, you should use this rule, that adenine 2 hydrogen bonds with thymine and C triple G. Okay, 3 hydrogen bonds. Alright, so this is the fifth type of numerical which could be asked. Now let us move on to solving questions. So here they've given is a gene coding for flower color has 5.6 kilobases. If number of cytosine molecule in this gene is 1000, then what will be the number of thymine molecule? So again, this is related to the fourth type which we had discussed now. So amount of adenine, guanine, thymine and cytosine all equal. So these are the all different types of bases present in DNA, right? So totally it has got 5.6 kilobases, which I can write it here. Alright, and we already know that adenine, since adenine double bonds with like double hydrogen bonds with thymine and C triple G, right? And both of them equal. The amount of adenine equals the concentration of thymine, and the concentration of cytosine equals that of guanine. So with this idea, I can write is I can replace adenine with twice of thymine plus guanine with twice of cytosine. Yes, this thymine, I have not replaced thymine with adenine because in the question they've asked the number of thymine molecules. So this is our required variable. So which is given to be 5.6 multiplied by 10 power 3 basis. Convert kilobases to basis. Now, you know that you can take common, right? 2 is common, so I'm going to divide 5.6 divided by 2. I bring this down here, and when I divide I get 2.8 into 10 power 3. Okay, so because in the question they have given the amount of cytosine molecules is 1000 bases, that's why I've converted the kilobase to base here, and here it is what, 2800. So the amount of thymine would be 1800. So this is the solution. Very simple. Alright. Just, every concept is already taught to you. Now let us go on and solve another question.